Today I'm going to show you how to access the data files from the experiment that you harvested on Monday the 21st of August. These are the data that you'll use to write your experimental report. So first of all, log on to Views. This is the welcome page. Go to Assessments, and it's Assessment 2, which is the experimental assignment. Open this window, and this gives you a bit of detail about when the report's due, what sort of things to include in it, and the kind of criteria that will be used for marking it. Now, what I've done is tidied the data sets up a little bit and transferred them to Excel files, which are a lot easier to work with. Because G11 results are in this link and G15s are in this link. Just write the report for the classroom that you're in. You don't need to do them for both rooms. Okay, so opening up G11, just save that. So once you've downloaded your Excel file, you're ready to open it up. So this is for G11. Now the first thing to notice is I've just included the final sheet for the entire classroom basically, so we don't have these separate groups anymore. Probably the first thing to do to have a look at the data is to freeze a pane. So this basically makes certain columns and rows static. So if we do unfreeze and then freeze that pane, so we can move along all the time, and you can see that these stay static. So we can look at the different responses, but at the same time we can see the labels basically. Everything has this safety column, so at any stage we can sort of go back and resort everything. So if I just do sort, safety, smallest to largest, it goes back. So you can see that this is all the results for group 1 here, with plants from 1 to 12. Now, as you know, a lot of our insects went missing, so we don't have values for their performance. We do have enough though, so this is good. And performance, what I've done is we've called this relative growth rate because we took some insects off the plants after 7 days and some after 4 days. So essentially this value is a change in herbivore mass between the insect going on the plant and when you removed it from the plant, or we removed it from the plant, and then divided by the number of days. So this would be, this value here, for instance, would be this value divided by 7, whereas this value, because the insect had only been on for 4 days, will be this value divided by 4. So relative growth rate is given as grams per day in body mass that's accumulated. So we're now ready to explore the data a little bit. And a really good way of doing this is to create a pivot table. So if you press this cell here, that actually just selects everything. It's quite a useful function there. And then go to Insert, Pivot Table, and you'll get this dialog box. So it's giving you the data range. It's asking you essentially, where will I find the information to calculate things like means? And because we've already got these cells selected, it's done that already. It then asks you where you want to put the pivot table. Now, usually it's just easier to put it on a new worksheet, so click OK. So this is the screen that you'll see. So you can see that there's now another sheet in the Excel worksheet. Now, each pivot table here gives you the option of categorizing what you want to look at. So obviously we've got things like silicon treatment. So you can see that we've got water only, solid silicon, and liquid silicon. If you wanted to look at a difference between herbivores, we have herbivores present, that's yes and no. Now what we can do is look at a whole range of different responses that we're looking at. So let's say we want to look at plant mass. We actually put it into the values field here. Now, by default, it just gives you the count, so the number of values that are represented. So it should roughly be about 30, but for some of you, your plants died, so there was no result there, so there's no entry. So for instance, 2 plants are missing here, 1 plant is missing here. Now what's good about these pivot tables is we can change these values. So if we click on this response, we go to value field settings, and it gives you a whole range of different things. So you could have maximum, minimum value. But probably the thing that we're most interested in is average, so the mean. So click on average. And what this does is it calculates the average value for plant mass under these different silicon treatments in the absence and the presence of an insect herbivore. Now the good thing about these tables is you can just do several of them. So I've just pressed copy. And you can see that exactly the same responses are in this dialog box here. So if I wanted to look at something else, let's say relative growth rate, again it will go to count, and you can see that in this column there's no herbivores, so obviously there's no values here. We should have a maximum of 30, but of course a lot of our insects won't miss him. So if we look at this again, go to value field settings, change to average, and we can see the relative growth rates for the different treatments essentially, and they're actually quite similar in this instance. A bit more complicated is calculating the standard errors, but you use this to give error bars on your graphs. So let's say we want to do it for average relative growth rate. What we do is we copy this pivot table twice. So this is exactly the same values as we've got here because it's still set on average. But what we can do is work out what's called the standard deviation. So go into value field settings and go to standard deviation. Now to get a standard error value, we also need to know how many observations we actually made. So we just change this one to count, which is the default setting anyway. So that tells you that there are 18 observations from the liquid silicon, 21 for solid silicon, and 18 for the water-only treatments. So to get the standard error, we calculate this by dividing the standard deviation by the square root of the number of observations. So in this case, this would be this number here, so that's E12, divided by the square root, which is all this means, of this value here, which is H12. Okay, so we can just copy and paste that. So it should go to the corresponding values. So yeah, E14 and H14. And you use these values to create your error bars.